Hi. I am excited to introduce to you Ryabhatta, an R-Shiny application that can be used to visualize, explore, and analyze single-cell RNA sequencing data from a variety of platforms. This application uses Sera in the background to perform data analysis and visualization. It works on existing Sera files that are present in your R environment. Let's get started. Select a Sera file in your R environment from the drop-down menu and click Load. This will set this file as the focus of the app's analysis and load features of the Sera, including a dimensional reduction plot, a cell count per cluster, and metadata on different panels. On the top left, all the genes in the single-cell data that are available for visualization are listed. You can select multiple genes to perform visualization. Once you have selected the genes, you can select the type of plot from the Plot drop-down menu. Then click Plot Expression. The plots for each gene appear as a separate panel under the dimensional reduction plot. Here you can see a panel of violin plots showing expression of each gene in all the different clusters. You might have noticed that some clusters appear to not have a violin appear for the expression of the gene. This does not mean that the cells within the cluster do not have any expression of the gene. To check if a few cells within the cluster express the gene, you can select to add show points in the violin plot and click Plot Expression. This will add points to each violin plot showing the expression of the genes in individual cells. Plotting time depends on the number of cells and the size of your Sera file. Here, we are dealing with about 10,000 cells. Now, even clusters that appear to have no expression previously begin to show a small population within the cluster showing expression. You can also choose other kinds of plots from the drop-down menu, like the heat map. In the heat map, the z-score transformed expression for each cell within each cluster is shown. Currently, you can plot up to 50 genes for any of the different plot types. A ridge plot shows the same information as a violin plot but is more compact. A dot plot shows the average expression of each gene in each cluster. The size of the dot shows the percentage of cells within the cluster that are positive for the gene. A color guide above each gene shows the average expression color for that gene. A feature plot shows the expression of individual genes overlaid on top of the dimensional reduction plot. This is the most common and popular way to show gene expression that is specific to a cluster. I have made sure each of the plots created is equal in size. So when you save the plots, they are ready to use in a publication. An expresso plot, short for an expression orientation plot, allows you to compare the expression of different genes. It overlays their individual expression into a merged plot. Up to 3 individual genes can be compared using this approach. The expression of each of the genes can be oriented with respect to the other 2 genes. This will be handy when you want to compare transcription factors and their target expression. The metadata added to the Sera files can be used to separate the conditions, or samples or technology used to generate the data. A separate app, that can create such Sera files from raw sequencing output files, and add metadata, is under development. When you plot the expression of genes with the metadata selected, the plot is split into individual categories showing expression per condition in each cluster. The violin plot is not the best for this specific data, because each cluster came from a different technique. Here, points do not align with the plot because of that. But, some clusters where the data is available from the different technique are aligned well with their points. As Sera is not specific to any unique single-cell technology, the app can be also applied to data from any of these technologies. Maybe we should try a different kind of plot to visualize this metadata difference. Let's see how the feature plot looks. The feature plot split on technique seems to make the point that different clusters show cells that were obtained from different technologies. I hope that the variety of plotting functions provided in this app will make it possible for people with minimal or no coding experience to jump into the visualization of data from single-cell sequencing projects. Next, we are going to look into the cluster naming and classification functions that are available in this app. The first kind of classification or naming tool is the graphical selector tool. This selector tool uses the feature plot of a gene to help you define a set of a cell population. Once the feature plot appears, you can click and drag around the cells that you wish to rename or classify. Do not worry if you did not select all the cells you need to select on the first try. Make a new selection. You can add a name to the selected cells. Currently, no space or special characters except an underscore is allowed for cluster names. Once you define a set of cells with this tool, the cells are renamed as such, and the change is reflected both in the dimensional reduction plot, as well as the cell count chart. Cell count tool tracks metadata selection and shows the distribution of cells amongst different conditions. If you wish to rename an entire cluster, you can do so using the second tool. Here select the cluster you wish to rename from the drop-down list and give it a new name. Again, there cannot be space or special characters in the cluster names. Once again, the selected cluster is renamed and is shown in the dimensional reduction plot. If you accidentally misnamed a cluster, or if you wish to restart naming clusters, you can choose to do so using the ident version tool at the bottom. Simply, select a previous version of the ident version and then, click Change Cluster Names. This will revert the cluster names to the previous version. You can also come back to the latest version that you named. Next, we will see how to use Ryabhatta to focus on a specific subcluster to conduct future analysis. Select the cluster you wish to focus on and select the number of dimensions you wish to use. The selected number of dimensions will be used to perform dimensional reduction on the subcluster. This will subset the data, normalize, scale and perform PCA analysis using the given number of dimensions. It will also run a series of clustering steps with various degrees of resolution. The greater the resolution number, the more numerous are the clusters identified within the subcluster. The cluster tree diagram shown below the dimensional reduction plot helps to visualize the increasing number of clusters with increasing resolution. You can play around with the number of dimensions you wish to use, and see if you are able to get a different spatial resolution of different clusters. Once a desired dimensional reduction plot is obtained, you can use the cluster tree diagram to choose the resolution. You can choose a resolution at which a reasonable or expected number of clusters will be obtained. Then, click Create Subcluster File. This will create a new SIRA file with the subcluster as the focus. Note that the subcluster has undergone a second dimensional reduction and therefore may not appear to retain the shape or position of the initial cluster. This is normal, and helps to show cluster distribution with a smaller group of cells. To aid in classifying the newly defined clusters within the subcluster, you can use the Find Markers option. There are 3 input choices to choose in selecting the markers. The minimum percentage of cells, within each cluster, that a marker gene needs to be detected. The minimum threshold and the difference of marker expression between clusters, and the type of statistical test you wish to use to define the marker. Once the markers are identified, a heat map of the marker gene expression is shown in the plotting area. This gives you an idea of whether a marker is exclusive to a specific cluster or is only marginally associated with a cluster. The name of the marker genes is provided in the table at the bottom of the app. At this point, you can choose to click on the Switch to the Marker View button. This will remove the dimensional reduction plot. If you now click on the gene name on the table, a feature plot with the gene expression of the marker is shown on the top. You can use this to evaluate the strength of the marker in defining a cluster. At any point during the analysis, if you wish to download the cell count data to plot it in a different way, you can do so by clicking the Download Cell Count button. This will download an Excel file with the cell count used to create this plot. The newly created SIRA subcluster enjoys all the plotting options of the original SIRA file. The app will also allow you to perform differential expression analysis based on any of the metadata conditions. Select the metadata to perform the differential analysis, and choose the categories of the condition that you wish to compare. The output is an Excel file that can be downloaded. You can also use the app to determine the cell cycle status using a cell cycle scoring approach. This will give you a bar chart showing the number of cells in each of the cell cycle phases. You can switch to percentage mode to compare between clusters. Finally, the app will also allow you to select cells that have a specific level of expression of a gene or a group of genes. Enter the gene names that you wish to use and select the range of expression. You can do this process iteratively, in order to select the cells of interest. As with any other classification, you can also add a name to these cells and analyze them as a group. Many thanks to you for watching this video. I will highly appreciate it if you could take a moment to share it with a person you think might be interested in this app. If you would be interested in using this app, please email the link in the description.